Take a look at the pour plate method for isolation. We may or may not get to this in the lab, but you should at least be aware of it. So the pour plate is different from the streak plate in that we are diluting each tube of media and then adding the same amount of media to different plates. And I'll show what that means in a little minute. But what we're doing is we are taking our culture, putting one loopful into the loop 1 tube, swirling it around between our hands, taking 2 loopfuls out into loop 2, swirling it around between our hands, taking 3 loopfuls out of 2 into 3, and swirling it around, and then pouring them into 3 plates, loop 1, loop 2, and loop 3. So what's happening here is that we have our little loopful of culture, right? And we're putting it into this first tube, and all of a sudden, we have that tiny amount in this 10 milliliters of BHI agar. Then we swirl that around and that spreads it out, and all the cells that are in there are now spread throughout the entire tube, ideally, right? Now, when we take a couple of loopfuls out of that, we'll be bringing over a lot less bacteria because we're only bringing a tiny amount of this 10 milliliters. So, into tube 2 will go a lot less bacteria. And then we swirl that around, and then we put 3 loops into loop 3, and again, we've diluted both of those loopfuls into this whole 10 milliliters, and so we have a whole lot fewer cells going into our loop 3 tube. So what happens then is as we pour into these different ones, we should see that somewhere in here, probably in loop 2 or loop 3, depending, we'll see isolated colonies. Now, this is important for a few things. Mostly, it's important for when we are trying to isolate obligate anaerobes. So, bacteria that cannot survive in oxygen, right? With the streak plate method, it's on top of the agar, and therefore it is exposed to the air, and anything that cannot survive in the air will die. With this, because it will be mixed in with the agar, it will be able to grow, and hopefully we'll be able to see some colonies there that are separated. Now, a couple of things to know about this is that, yes, these are tubes of agar, and they are melted, but they can't be too hot or we will kill the cells. Remember, agar solidifies at about 40, 45 degrees Celsius, right? And that's right there at the top of where something like E. coli can really survive. You can push it to 50 degrees, but there's not a whole lot there. So we need to cool these down until they are cool enough that we can comfortably hold them in our hand for 20, 30 seconds, something like that, without having to put it down. And again, that depends on how used to heat your hands are. But once you've done that, you're able to proceed. We are not quite there with this, but I'm going to go ahead and fake it anyway, just so you see how this works. So we have our sterilizer, and we put our loop in there. When you're not using your loop, there are these little holes there. You can stick your loop in there, and that will keep the loop itself from being on the bench. All right, so we sterilize our loop. Before we do that, we want to loosen the tops of the tubes, again, like always, just that we can get there easily without having to wrestle with everything. All right. So our loop is sterilized. We grab our culture. Remember how we do it. We pick it up like that. We swirl. We go in. We go out. Now, first tube. We put it in. We swirl it around. Now, we take 2 loopfuls of that. We swirl it around, sorry. And we take 2 loopfuls of that, put it into our second tube, right? Then we swirl that around to mix everything up. Then we take 3 loopfuls of that, into our third tube. And ideally, of course, you don't want to touch the tops of that. Now, we don't have a lot of time because remember, this is close to settling temperature already. So we need to have our plates ready, and we have our tubes ready, and we go. Again, you just crack it enough, and you pour in your agar, you swirl it around, you set it down. You grab the next one, which ideally, again, you'd want to have the lids on there, but pour it in and go. Grab your loop 3, pour it in, and go. You'll notice that there's not nearly as much in here as when you poured the plates, but there's enough that you should be able to get some growth inside the agar. I know it doesn't look very deep, but when you're a bacterium, that's like kilometers deep. So that is how you do a pour plate. You then wait for them to set, then you put them in the incubator, wait for 24 hours or 36 hours, depending, and you see what has grown, and you're able to do that to hopefully isolate some bacteria that will not be able to survive in oxygen. And that is the pour plate method of isolation.